This is a production of Cornell University.
Today, I will talk about the orange gene function in nutrition quality improvement, and also our recent finding about the orange gene function, potential function in creating climate resilience crops. So first, I'll introduce the orange gene function in crop nutrition improvement, mainly for carotenoid, and also our recent finding about the OR gene regulates chromoplast division. And then I will briefly introduce our efforts to increase carotenoid, stable carotenoid accumulation in seeds. Finally, I will talk about our recent findings in the critical role of our orange gene in photosynthesis. So I'm a research associate in Dr. Lily's lab. So our lab mainly works on crop nutrition. So orange gene was first cloned in cauliflower. So in cauliflower, it's a transposon insertion, which caused different splicing of orange gene, and finally can induce chromoplast formation and a huge amount of carotenoid accumulation in cauliflower curds. Later, we find that in melon, a golden snip is responsible for the carotenoid accumulation. Like a cantaloupe we in our market, so which also contains the golden snip. This golden snip caused amino acid substitution, changing from arginine to histine. So that's OR histine. So we find that OR histine is responsible for the induction of chromoplast formation. So in recent years, several other groups also find that OR gene is associated with the major QTL of carotenoid accumulation, like in sweet potato, in carrot, and also in cassava. So those OR genes may contain different kind of mutations, which also responsible for high amount of carotenoid accumulation. So this makes OR gene a very good candidate for the crop nutrition improvement by breeding. So our lab and also other lab also use OR gene as a tool to manipulate carotenoid accumulation by overexpression OR in potato tubers can increase carotenoid accumulation. We also introduced OR gene in tomato. Normally in tomato at the early stage, the tomato fruit is totally green. But when we introduce OR gene, it can induce chromoplast formation and carotenoid accumulation at the very early stage. Also, by introducing OR in combination with the carotenoid biosynthesis enzymes, we can get high level of carotenoid accumulation in rice. So with the advancement of the genetic of the CRISPR-Cas9 genome editing method, in future we hope to precisely edit the OR gene to OR his in different kind of crops to finally improve the crop nutrition content. So as we all know that plastids are essential plant organelles, which there are different types of plant plastids, including the aminoplast, which is the main site for the starch accumulation, and also the chloroplast is the site for photosynthesis, and the chromoplast is the main site for carotenoid accumulation. We find that with the OR mutant or OR his gene, there's only 1 or 2 large chromoplasts in one cell. But in tomato, there are many chromoplasts which lead to a high level of carotenoid accumulation. So when we do the OR his callus in orthosis, we find that it can still induce chromoplast formation, but only 1 or 2 large chromoplasts in one cell. So we are thinking that that's the limitation of the carotenoid improvement. We are thinking that if we can increase the chromoplast number, so we can increase the carotenoid accumulation sink capacity. to finally increase the carotenoid level. So we want to figure out that why OR hits limited the chloroplast division. So we find that when we check the Y type OR his GFG localization in non-green plastids, we find that OR is associated with the division ring, which is in the middle of the plastid. But in chloroplast, we find that OR the localization of OR is different in non-green plastid. So that implies that OR in non-green plastid or chromoplast may have different function in chromoplast division. So when we do the Z-axis scanning, we can clearly see that OR is more concentrated in the binary fission site, localized in a ring structure. So in OR hits, we can only see 1 or 2 chromoplasts, but in OR Y type, the plastid number is normal. So the question is how does OR regulate chromoplast duplication? We want to figure out the molecular mechanism. For chromoplast division, there are no many reports about that, but for chloroplast division, it's a well-established mechanism. There are different rings, inner rings, outer rings. They constrain the chloroplast, and finally, by the binary fission, the chloroplast divided into 2 separate chloroplasts. So then we check the interaction between OR hits with those chloroplast division factors, and we find that OR hits can interact with AX3, which is the important division factor for chloroplast division. Normally, AX3 will interact with PX6, but with OR hits bind to AX3, it will compete with the PX6 to bind AX3. So we proved the interaction by ester 3 hydride that in the absence of OR hits, AX3 can interact with PX6. But with OR hits, AX3 will mainly interact with OR hits. We checked this interaction by effect, and finally we want to prove the interaction in vivo. So we designed the co-immunoprecipitation experiment. We express both AX3 in both OR-Y type and OR-his background, and find that if we express AX3 in OR-his background, AX3 will mainly bind to OR-his instead of PX6. So then we prove that OR-his may interfere the PX6 and AX3 interaction to affect chromoplast division. So since we know the mechanism of how OR-his affect chromoplast division, then we want to correct it to make a more chromoplast in OR-his background. So we try to overexpress different kind of plast division factors, the FTS Z1, Z2, PDV1, PDV2. Those are all division factors that affect chloroplast division. So we want to see that which one can increase the chromoplast number in OR-his background. And finally, we figure out that when we overexpress PDV1, we can increase the chromoplast number in OR-his background. So which makes the callus accumulate more carotenoids in the chromoplast. So for this project, we figure out the non-green plastid, especially a chromoplast division mechanism, which is less studied in previously. And also, this research provides a potential mechanism to increase carotenoid content by increase the chromoplast number instead of the biosynthesis ability. And also, there are still some questions we need to answer is, what's a major duplication mechanism for chloroplast? Like in different crop, some chloroplasts, they are transformed from chloroplast. So they may use different mechanisms. Some chloroplasts may develop directly from proplastid. They may use a different, like binary fission or the bundling mechanism. And we only, for this study, we only tested the interaction between PX6 and AX3. So we still don't know that is there any more factors involved in the chromoplast division, especially when ORH is not existed. Then we already know that ORH is a great tool for carotenoid accumulation for crop nutrition improvement. We also know that the final nutrition content is the bioavailability of the nutrition content is very important for our crops. We know that carotenoids are not very stable because they have several double bonds and they are very easy to be oxidized. Especially the seeds during storage, the decay of the carotenoid, vitamin A carotenoid is a serious problem. So we want to increase the carotenoid content in seeds. Additionally, we want to, the carotenoids in seeds are more stable during storage. So we investigated the carotenoid biosynthesis and degradation gene expression, and we have a general idea that we want to increase the carotenoid accumulation in seeds. Meanwhile, we want to decrease the degradation and also fight against the oxidative stress in the seeds during the seed storage. So for this project, we will target several genes. For ORHs, we target ORHs, we overexpress ORHs to increase the stable storage of carotenoids. And we overexpress PSY. PSY is the photosynthesis, is the bottleneck for carotenoid biosynthesis. We want to increase the PSY level. And also we need to knock out BCH2, which is the beta-carotene hydroxylase, which can degrade carotenoid. And additionally, we overexpress the HGGT, which is for vitamin E biosynthesis. Vitamin E is very important because of its antioxidant activity. So we want to increase vitamin E level in seeds to reduce the oxidative stress and make carotenoids more stable during storage. So we designed several constructs with BCH2 knockout, and overexpressed PSY, knockout BCH2, overexpressed PSY, and also overexpressed ORHs, and a large construct targeted 4 genes, knockout BCH2, overexpressed PSY, or overexpressed ORHs, and also overexpressed HGT. So the result looks great. So we observed that the seeds at the green stage, they already start to accumulate a high level of carotenoids. When we take a close look over the seeds, we will find that we can already see the chromoplast formation in the young seeds. And also we did the carotenoid stability experiment. We figured out that the carotenoid stability are largely enhanced because of OR and HGGT after 8-week storage at 37 degrees, which in wild type, the carotenoid, they only have less than 50% left after the storage at this condition. So this, although this experiment is conducted in Arabidopsis seeds, but it can be adopted for other crops, for other seed engineering to increase the stable carotenoid storage in seeds, and therefore increase per vitamin A content. So all these experiments, we are talking about the nutritional aspects of OR gene, especially the OR his, the mutant version of OR. Then there are, because OR is a highly conserved gene in green plants, there are a lot of plants they don't need to accumulate a high level of carotenoids in fruits or in flowers or in roots like carrots. So what's the function of the wild type OR gene in green tissues? So that's a question we always ask ourselves. So we generate OR overexpression lines and OR like double mutant lines, knockout lines, and we figure out that without OR, plants show the pale green phenotype, and with significantly reduced photosynthesis ability. And when we check the chloroplast structure, we find that without an OR gene, the cycloid memory stacking is missing. So that makes the chloroplast cannot function properly. When we do blue native page to check the photosynthesis protein complexes in chloroplast, we find that there are several major bands missing in the blue native page, like the light-housing complex assembling the photosystem 1, photosystem 2 dimer, and the silver complexes for photosynthesis. When we check the proteomics data, we find that there are many proteins in the light-housing complex are significantly reduced because the knockout of OR-like gene. So we also did the second dimension Western blot of the blue native gel of each light-housing complex proteins. We find that there are many major proteins, especially light-housing complex protein LHCb1b2. They are missing in the light-housing complex assembling to a large complex. So previously, our lab showed that OR is the molecular chaperone of PSY, phytosenthesis protein. Is that the reason OR like mutant has less photosynthesis complex? So when we check the chlorophyll biosynthesis pathway, we find that OR like double mutant can accumulate protoporphyrin 9, which is the intermediate of chlorophyll biosynthesis. Normally, we will not see protoporphyrin 9 accumulation in wild type plants or in other pale green mutants, except for the enzymes directly convert protoporphyrin 9 to mechanism for protoporphyrin 9. So this result indicated that in OR like double mutant, not only the carotenoid biosynthesis is impaired, the chlorophyll biosynthesis is also affected. So we checked that because OR is the molecular chaperone, we checked that if OR also function as a molecular chaperone of chlorophyll biosynthesis enzymes. And finally, we figured out that OR directly interact with the mechanism catalysis subunit I. So mechanism catalysis is several enzyme complex. There are several subunits, CHLI, CHLD, and the CHLH, which is also named GAN5. We confirm that OR directly interact with CHLI. Then we checked the protein stability. We figured out that in OR like double mutant, the stability of CHLI and PSY is affected. So their turnover is very quick. And then when we check, because the mechanism catalysis is a multiple subunit enzyme, we checked that if CHLI assembling is also affected. Our results show that in OR like double mutant, CHLI cannot assemble into larger enzyme complex. Then, since OR is the molecular chaperone, it can protect the protein from denaturing. So we checked that if we use higher temperature, which may cause faster protein degradation. Previously, people reported that CHLI can be significantly affected by higher temperature stress. So when we do the heat stress treatment, we find that when we overexpression or when in the overexpression line, CHLI and PSY showed higher stability than wild type. And then when we do the enzyme activity assay, we find that when we treat magnesium chelatase with higher temperature, so the enzyme without OR showed a lower activity compared to the enzyme complex with OR. We got a similar result with PSY. In OR overexpression line, PSY showed highest activity, but in OR double mutant line, the PSY activity was significantly reduced. So that makes us think about that OR may regulate both carotenoid and chlorophyll. Both carotenoid and chlorophyll are photosynthetic pigments. So the right amount and the right content of those photosynthetic pigments are very important for the proper function of plants. So we generated OR overexpression line, and also we have OR overexpression in tomato line. We did a heat stress treatment. We find that OR overexpression line in arthrosis showed a higher thermotolerance. So when we check the pigment content, we figure out that overexpression line, the pigment content are less affected by the heat stress. So the similar phenomenon was also observed in OR overexpression line in tomato. So the OR overexpression line in tomato, when we do heat stress treatment, we find that the OR overexpression line lines are less stressed because they accumulated less anthocyanin. Anthocyanin is a kind of indicator of the stress response. So the white type tomato leaves, they accumulate a large amount of anthocyanin. Also, the pigment content, both chlorophyll and carotenoid, they are also less affected in tomato or overexpression lines. So for this experiment, we figure out that OR is the master regulator of both chlorophyll and carotenoid biosynthesis pathway. And OR is the chaperone of the first committed steps for carotenoid, that's PSY, phytoin synthesis. For chlorophyll biosynthesis, OR is the chaperone of CHLI, which is a part of magnesium chelatase. So as a chaperone, OR protects both pathway and against the stress condition. So that reminds us that the function of OR in plant resistance to abiotic stress. So actually, there are already several reports about the overexpression of OR in to increase the plant resistance to abiotic stress, but the mechanism is still unknown. So our this study provides a mechanism explanation of why OR can increase the plant abiotic stress resistance. So finally, we conclude that OR can not only function in the crop nutrition improvement by manipulation of OR expression. It can be a good candidate for the crop for generate the climate resilience crops to increase the plant resistance to abiotic stress like heat stress. So that's my talk today. Thank you for our lab members and our collaborators. This work is funded by USDA RS funding and NIFA funding. I'd like to take any questions. Thank you. Yeah, that's a good question because that's possible that carotenoid can provide precursor for ABA. So ABA might affect the plant resistance to stress conditions. So that's just one part of our explanation of our chaperone function. For our Y-type overexpression, we didn't see the huge increase or huge improvement of carotenoid. So that might be not the main reason of the anti-stress or antibiotic stress of our function.
What is primarily actually in the carotenoid? So how it is linked to the anthocyanin? Like high anthocyanin content is overexpression, right? So poor.
For this experiment, anthocyanin we just used as a kind of stress indicator. So in many kind of stress conditions, we will see the anthocyanin accumulation. So for this part, we just indicated that plants are less stressed. So we didn't see the direct connection between carotenoid or anthocyanin. But in fruits, like apple fruits, there might be correlation between anthocyanin and carotenoid accumulation. That's out of the scope of this study. Yes.
For your, I think, second project, when you talked about overexpressing OR-His and the chromoplast division factor, how did the size of those double mutants compare to the single big chromoplast that you had in the OR-His overexpression lines? If you go back to the slide, I can't remember the name of the gene.
PDV. We overexpress the PDV1 to increase the chromoplast. Yeah. Actually, we increased the chromoplast number.
Yeah. So how did the size of the higher number chromoplasts compare to the size of wild type or only or his mutants?
In this slide or in this slide? We overexpress the PDV1 to make more chromoplasts, so the number are increased.
So the size is comparable to the wild type chromoplast size then?
Yeah, the chromoplast size is comparable to the so for OR-His, it only contain one large chromoplast, but I think there's a balance between the size and the number.
Yeah.
So you cannot get the larger size and a lot of chromoplasts. But for this project, for this experiment, we observed the increased chromoplast size, but the increased chromoplast number, but the size are not comparable to the OR-His.
Okay.
In OR-His, just one large chromoplast. And we also observed that the size were slightly affected by the overexpression of AX3. I didn't show in this slide. Good question. Thank you.
When you're counting and measuring chromoplast in the study, do you assume that any process containing carotenoids is a chromoplast, or are you identifying between chromoplast and pneumoplast?
So because we are using the callus system, so under the light microscope, we can clearly see the chromoplast like this one. So we can clearly see the chromoplast instead of, so for other type of plastids like the aminoplast or the even proplastid, it can hardly observed under light microscope. But for chromoplast, it's easy to observe under the light microscope. Thank you.
Thank you.
This has been a production of Cornell University. On the web at cornell.edu.